forward on this. Okay. All right. We're going to attempt to go through lecture 1 to 3
Okay.
today. Are you able to stay the whole time, or were you planning on cutting, or are you just going to see how it goes?
We'll see how it goes.
Okay. All right. Because
Yep.
if you feel like if it ends up only being you and I,
Okay.
honestly, like if you're feeling like I'm done or my kids need me, just don't worry about it. We'll just pick up another time. So no stress.
Yeah. We are going through like 4 and 6 another time. Hopefully I can make it too, right?
You have a 4-year-old and a 6-year-old?
No, I meant chapter 4 and 6, right?
Oh, yes. Yeah. Yeah. I'm going to
Now
try.
till Sunday.
Yeah. I'm going to try to do another study session. I also have an anatomy study session that I do on Sunday nights. And so I'm trying to cram it all in,
Okay.
you know.
We'll just do the best we can.
All right. I'm going to go to slideshow presentation. Can you see that?
Yes.
Okay. Some of my animations might be wonky, but it's okay. So this was my intention to have everything here in one place so we can just go through it all, but we're only going to do lecture 1 on this Google slide PowerPoint presentation. Then I'm going to just switch over to Dr. Dan's slides and we'll just talk about it. So let's see. Okay, we'll start with this. What I thought is I would just throw some questions up and then we can just talk about different things. So this is just an overview of the very first PowerPoint presentation that we were given. So this whole section will cover what is microbiology. We'll talk about prokaryotes versus eukaryotes, microbial diversity, just like bacteria, fungi. We'll talk about like all of the characteristics of each one of these microbes. So it'll be a good refresher because I think it's been a while.
It's been a while, yeah.
Yeah. Beneficial microbes versus pathogens, history of microbiology, and taxonomy. Like if you remember how we name things, like remember how we kind of talked about like kingdom, phylum, class, order, family, genus, species, that kind of thing, and how we name microbes.
Okay.
So we'll start off with prokaryotes versus eukaryotes. There was like a few distinguishing factors that was like separating these two things. I know I'm not using the right terminology, but do you remember what some of the differences between a prokaryote and a eukaryote are?
One's no nucleus and eukaryote has a nucleus.
Good.
Prokaryotes is 70S.
Yes.
For DNA, or is it RNA? DNA, right? Or
ribosomes.
Ribosome, sorry. And
yeah, you're right.
Prokaryotes doesn't have organelles.
Yes.
And eukaryotes have all the other organelles, if I'm correct.
Yes. Yes. Okay. Yes. And then one of the other things he said was that prokaryotes have circular DNA and eukaryotes have segmented genomes.
Okay. I thought is it linear? Okay. So it's the same.
Yeah, linear. You're right. And then there's one copy for prokaryotes and then multiple copies for eukaryotes. And then you said no organelles for prokaryotes, organelles for eukaryotes. 70S, 80S. Oh, right. So all prokaryotes are unicellular.
And these are multi or uni?
Yes.
Organism. Okay.
Yeah. So, and this is the whiteboard picture that he drew. I just wanted to include that because I know he had mentioned that whiteboard notations are all fair game for exams. And so he just kind of drew a picture of what a prokaryote looks like, no organelles, single copy, circular chromosome, and then
segmented chromosome.
And so, yeah, I remember he was saying something about like our DNA, we have 12 pairs, right?
23?
23.
I was like, what?
What is it again?
I don't know.
That's why it's good not to study by yourself, because I would be wandering out in the wide world saying wrong things.
I was like, wait, what class? But prokaryotes has one, only one. Okay, I gotta remember that.
Yes, single copy. Yes. And then they have circular DNA, like circular. Yeah.
Okay.
Okay. One of the things that Dr. Dan says that really helps kind of trigger some of the information is just say everything you know about viruses, and then kind of study the stuff that you don't remember. So what do you remember about viruses?
Well, I just reviewed lecture 6 like literally 2 hours ago. They're not prokaryote or eukaryote.
Yes.
That's one thing I know. And then they have a capsid that protects the DNA or the genome.
Yes. I'm impressed already.
And the one that doesn't have a membrane is naked, of course, because it has naked. And then there's these spike that it sticks onto a certain receptor. And then the one with the membrane, which is from the host, that will have their spike out on the membrane, but they are very sensitive to temperature. So certain medication can break down that membranes, like soaps and stuff. So that's one thing.
That's good.
Let me see what else did I
Way
remember.
more than what I remembered.
I'm really bad under pressure. Then I forget.
So this is one thing that they said, like in our notes, that
yes,
you said they're neither prokaryotic or eukaryotic. That's right. But he wants us to classify them as obligate intracellular parasites.
I need to remember that.
And I have written that down many times because I know that I'm going to forget that. But I for sure don't think I can recall that. So I'm going to need to write that down again.
Same thing.
So a fill in the blank here is viruses are dependent on what for their activities?
A host.
Yes. Good. Look at
They
you.
would not survive without a host. Well, they're not effective, I guess, without a host.
Yes.
Or to divide, right?
Yes.
Right.
The majority of microbes are free living and get their food from. So this is not virus related, obviously.
Okay.
So they're free living out in the wide world and they get their food from.
Can I get a hint?
I just put it down. Their environment.
Okay. I was thinking host too, but I was like, you can't ask the same thing.
Right. So I guess if they're free living, they are just out getting their food however they get it. They're not like, as opposed to being a parasite where they're attaching on to something and getting their food from the host, right?
That makes sense, yes.
True or false, microbes are multicellular.
Well, they can be either or, right?
Mm-hmm.
So that's a false.
Mm-hmm. They are unicellular.
Okay, good.
Multicellular. I know. I kind of worded that funny where I was like, it's half true.
So like it could be true, but then there's like what's the other one that's unicellular? Which one is it?
Oh, unicellular? Is it bacteria?
Is bacteria the only one that's unicellular? I thought there was one.
No, there's also is it algae? Algae can be
algae
no.
is both. I think algae is both.
Fungus. Fungus can be unicellular and multicellular, I think, because it comes in both yeast and mold.
So
yeast would be unicellular, mold would be multicellular.
What was that one that's unicellular? I have to figure that one out. Yeah, I'm already forgetting things already.
I think it's actually going to come up when we talk about the characteristics of each microbe. Okay, this is just review vocabulary. I hope I animated this okay. the definition of prokaryotic cells. So I basically took everything that was in the PowerPoint that was kind of highlighted. I'm like, okay, I'm going to call that vocabulary so we could just talk about that. So
Mhm.
just identifying prokaryotic cells. I copied and pasted this, so I don't have this like readily in my brain.
That's fine.
Um,
I don't either. You can tell.
so I would say it would be some of those general characteristics that we talked about, like prokaryotic cells don't have a nucleus, they don't have organelles.
The
Um,
Duckler, maybe that's it.
70S. Let's see if
And um one copy of their DNA.
yes, yeah. And it's circular DNA. So that's the first definition. Eukaryotic cells. So we remember those have a nucleus,
Mm-hmm.
have organelles,
Nails.
they're ADS.
ADS, they're linear segmented chromosome, 23 pairs of
Yeah.
chromosome.
Yes. I guarantee you I'm still gonna not remember that because I've heard that all my life, but it doesn't
When
stick for some reason.
you can remember.
I can recall it if I need to,
Yeah.
right? Like if I'm telling somebody about it, I'll just need to talk about it more. So cells that contain a nucleus and membrane bound organelles.
Remember. Okay.
We were just more specific. We were better than the definition. Organelles. Do you want to take this one?
It has, of course, ribosome, a nucleus, a nucleoid, because I think the other one has nucleoid, right?
Right. You're describing different kinds of organelles. But if
Okay.
you were to describe the definition of
Of
organelle,
an organ,
like how would you describe it?
a membrane
Uh-huh.
bound
Yeah.
structure.
Yeah. That has maybe like a specific function. That's what I would guess, maybe.
That's good. Hey, okay.
Internal compartments in cells that are surrounded by one or more membranes. Hey, you were spot on. That was
I
good.
just need to remember the word internal compartments in cells. Okay.
Yeah. I think you would be able to identify it though if you needed to. I mean, I think worst case scenario, if you saw that definition or it would be like a fill in the blank, you'd be like, oh, yeah, internal compartments in cells.
Mhm.
Like, what else could that be? It's got to be organelles.
Right.
Obligate intracellular parasites. We just talked about that.
It's a virus.
Right.
But to define that, oh, okay.
Oh,
Classification of
sorry. I
virus.
animated that too fast. I like clicked it too fast. So classification of viruses, remember, they're neither eukaryotic or prokaryotic.
Right.
And then macroscopic versus microscopic.
I think macroscopic is something that we can see visually with our eyes, and micro is too tiny that will require a telescope. I mean, sorry, microscope to be able to see them separately.
Right. Yeah, something that you can't see with your naked eye.
Oh, that's everywhere.
Yes, good.
I just don't know how to spell that correctly.
Right. But he sounded like he was like, hey, if you don't spell it right, I'm not going to mark you down for that. That's what he told us for the lab exam.
Yes.
Yeah.
Ambiguous. Okay. Free living. You just said that it's living out in their own environment.
Yeah.
A host.
Yeah. Yeah.
Okay.
They don't need a host.
Thanks. Reviewing this for me.
It's good to review.
Yeah, it's good because then I'll be able to remember like what you just said.
Right.
Parasite, something that will infect, pathogenic. I don't know.
Yeah.
One parasite.
And I think like another thing would be like they need a host.
They need a host depend on for their activity.
Yes. Yeah. I don't think I wrote this definition out, but that sounds good to me. Photosynthesis.
A chemical reaction where you get energy from sunlight.
Yes.
Okay.
I didn't write that one down either. Theory of biogenesis.
that living things come from living things.
Yeah. Yeah.
Life comes from a living thing or form.
Yeah. So
And
I like when what helps me is when I look at this, like bio is like living things, right? Like biology. And then
Genesis,
Genesis is beginning.
beginning, okay, life begins from living things.
Right.
Okay. And who was the person that is it past Louis Pasteur? I think that was the
We're
person.
gonna go through it, but the theory of biogenesis, life comes from life. Yeah, I think it's Pasteur.
Him, right?
Mhm.
Okay.
I'm pretty sure.
I'm sure that will be a question too
Yeah.
about who it is, because we have to
Yeah.
know about who that person is connected to a certain theory.
Right.
Yeah.
And we'll go over that. I've got some slides for that too, so we can go over that.
Okay.
Aseptic techniques.
A technique where you are killing anything that will cause contamination or
Yeah.
to decontaminate
Yeah.
materials.
Yeah. The aseptic techniques that were developed to like reduce microbial growth is what I think. Yeah. You could tell because I made these slides. So that one I just remembered.
Okay.
And the germ theory of disease.
I don't know
I
this
think I
one.
know this one. The germ theory of disease.
Germ theory. What exactly is? I don't know this one.
Many diseases are caused by microorganisms.
Oh, okay.
So if you remember that slide that had the flies on it, like you had the 3 different jars, and
Mhm,
they were trying, like, because I think the theory at that time was that um, life just kind of spontaneously happened.
mhm.
And I can see his face, but I can't think of his name right now. But he had the 3 different jars, and he put the meat in the 3 different jars, and one
Yeah,
of
I
the
remember. Yeah,
containers was
I remember his
open.
face.
Yeah. What did you say?
I'm trying to remember his name too right now. It'll get to us. Many disease caused by.
I know that aseptic techniques are Lister,
I
like
know that
of
too.
Listerine.
Listerine, yes. Oh my goodness. I was thinking the same thing.
I was like, I don't know why. That's probably
And they
gonna
have
take
to
forever.
swish with a mouthwash, which is aseptic.
Yeah, Listerine.
Now I have to think of this germ one to remember. Oh, let me see.
And then that germ theory of disease. Gosh, I can't think of his name. We'll talk about it. We'll go over it. Phylogenetic tree. Shoot.
Okay, this one I can't seem to find the words. It's just the way it's a way they name classification of different like groups and class, or I
Yeah.
mean it's the way of microbiologists or scientists name
Yeah.
a organism.
Yeah. Shows the genetic and evolutionary relationships between Earth's habitats.
Okay, gotta remember all that, huh? Show the genetic and evolutionary relationship. Okay. Binomial system of nomenclature.
So bi is 2. Nomial, I think, is name. So
Mhm.
2 name system of nomenclature. So it's like the way that we name. I think it's just organisms in general, right?
Mhm.
Living things in general. Assigning scientific names to organisms.
Okay, assigning scientific name.
Yeah. So like we had talked, I think, about like, like humans, like Homo sapiens.
Mhm.
So that's the system. It's called the binomial system of nomenclature. Okay, 6 types of microbes. These are the 6 different ones that we've been going over.
Algae, helmet. Is that 3?
What is the first one you said?
Fungi?
Fungi. And you said algae,
Protozoa,
helminth.
algae, helminth, protozoa,
Protozoan,
virus.
virus,
Right?
and starts. It's the one that you were. Oh no, I was going to say it was the one that you were just studying, but you just studied viruses.
Protozoa.
No, you said that one. I think you're missing bacteria. I don't think you said bacteria.
Oh yeah. Duh, that was the main one. Yes, bacteria.
We have fungi, helminths,
Hi,
bacteria.
Giselle.
Hi.
Hi,
Welcome.
Giselle.
I need your help.
Let's do it.
We're just going over lecture 1.
Okay, sounds good.
Okay, what are the general characteristics of bacteria? So they are, I think I started with some, let me just see. Are they prokaryotic or eukaryotic?
Prokaryotic.
Prokaryotic, good.
Unicellular.
Unicellular,
Free living. Okay.
They're free living. Some are photosynthetic,
Some are
some are
pathogen.
pathogenic.
Okay.
So this is definitely something that I have to like touch up on because I haven't studied this in a while.
Me either.
Okay, we'll go on to fungi. Are they prokaryotic or eukaryotic?
Eukaryotic.
Yes.
And they are multicellular. I always remember them as multi because of the yeast and mold, because
Yes.
yeast can be unicellular and mold. I just think of the bread and it's growing, so it has
Yes.
to be multi.
Right. That's a good way to remember it.
Yeah.
They're not photosynthetic.
Right.
Do you remember anything else about it?
They do external digestion
Mhm.
and saprobes, yeah.
Saprobes.
External digestion. Okay.
External digestion. So how do you guys remember how external digestion works?
It would spit out some sort of enzyme and then like the enzyme will break down whatever it's breaking down and then it'll just slurp it back up or something like that.
Good descriptive words there.
Thank you.
Giselle.
Oh my gosh, so funny. Okay, protozoan.
Every single one.
Is it right though?
No, I think you're right.
I just remember that it's like a fly.
Oh,
Yeah.
right. Yeah.
And I saw a terrible movie called The Fly where this guy turns into a fly, and I'll never forget that scene. So it's disturbed me forever.
What movie is this?
It's called The Fly.
Okay.
It was a terrible movie. Don't watch it. It's probably really lame now, but yeah. The graphics weren't up to par. The general characteristics of protozoan.
Protozoan are
Eukaryotic.
The animations will come later.
okay.
It's just how it arranged in the thing.
Gotcha. Okay. I think they are multicellular.
I think they're multicellular too. Yes, I think they're multicellular.
Since we just had a lecture yesterday, I should remember this.
I think they're unicellular, no?
Are they?
I don't
We'll find out when the animations come to life. And I think they're
Oh, he's right. They are unicellular. Yeah, it is. Well,
Internal digestion.
okay. And they're parasitic, are they not?
yes.
Yes. And free living or free living. It's both.
Right. Okay, that makes sense. Here comes the animations. They're eukaryotic. They're unicellular. That's what I have. Let me escape so I can just go back to the, I will go to the notes just to make sure that I have that down correctly, because I have been known to write things down wrong. I'm just going back to the lecture slides. We are on fungi? No, we just, they're unicellular.
Mhm.
Yes.
They're unicellular. I will go back. Are you guys still seeing my slideshow?
Yeah.
Okay. I'm gonna zip through these guys. Internal digestion. And I think maybe a good way to remember that, because I think of it being like parasitic, like it attaching to something and absorbing it rather than it having external digestion. I don't know.
Mhm.
Okay, algae.
You
Eukaryotic.
multicellular,
Mm-hmm.
photosynthetic. I mean, yeah, photosynthetic.
They're all unicellular or multicellular. Yes, photosynthetic. Good.
And always in water or
Yes,
associated with water.
good. Is that it? Let's see. Oh, I reanimated these. There you go. What are the general characteristics of helmets?
Eukaryotic.
They're eukaryotic. So everything is going to be eukaryotic except for
Bacteria.
viruses and bacteria.
And
right?
yeah.
Yeah. So they're eukaryotic.
Parasitic worms.
Parasitic worms.
Multicellular.
Good.
Yeah.
Else?
You missing something?
Nope, that was it.
Oh, okay. Only 3 things. Okay.
Yeah. That should say these scientists. I started panicking for time, so I didn't.
You need to learn how to spell his last name.
Right? I know. I was
Well,
like,
that's a
we
good
should
question.
do like a spelling test. Just like, we're going to need to remember how to spell this. 2 E's, U-W-E-N-H-O-E.
Okay.
Okay. What are the contributions of Antony something?
Then
Leeuwenhoek. 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 I think it's with a V.
Yeah.
He made a microscope.
yes, the father of microbiology.
Right. But he wasn't the first one.
Animalcules.
Right. He wasn't the first one to make the microscope. He was just the first one that made it so well so you can see.
Yeah. It was like, yeah, they had the best magnification. It was like 300 times.
Yes. Yeah. Good memory. Good.
He was the one that was like, there are so many women who are getting who are having children in the hospital and dying. And then everyone thought he was crazy. But then who was it? Lister? Was it Lister? Joseph Lister, who was like, you know what, this guy's onto something. And then he was like, you're right. And then the aseptic technique happened.
Yes. Good. And then we talked about Louis Pasteur just a little bit a couple minutes ago. Hoy, do you want to take this one?
Was he the one that you mentioned about the 3 jars with the magnet growing on top of it? Or he was no, he did that round.
I think it's in the name. It's actually in the name because the technique that he came up with is pasteurization.
He developed pasteurization.
Mhm.
The thing that he did though was the flask with the needle
Yes, yes.
thing or that straw or whatever it
That,
is.
yeah. He said that air has microbe. That's why living things will grow from living things, which is the theory of biogenesis, I think.
Genesis, yeah.
Yeah.
Yeah. Actually, he might be with the one with the flasks.
No, he's one of the flasks, but which
He's
one?
not the one with the fly with the red meat flask guy.
No, the red
That
meat
was
guy
someone
was
else.
someone else. Yeah.
That was the Ready experiment.
And then we
Yes,
just talked about-
Ready, his name, the
Yes.
red meat guy.
Yes, exactly. Ready, the red meat
Red
guy.
meat guy, okay. Thank you.
And I don't know that we talked about so much about Robert Koch.
Yeah, I think he said we don't need to know about them yet or not yet. He didn't mention much of it, but I don't remember.
Alexander Fleming, I had to rewatch this just on this segment, and he actually does mention a little something about Alexander Fleming. I'm not sure if we'll need to know it or not, but he called him the father of antibiotics. It'll come up in the animation after we're done with this.
Good to know, Father.
Yeah.
Okay. I didn't even catch that. Okay. Thanks.
Yeah. Yeah. I just rewatched it just to be like, okay, how many of these do we need to know? Because I remember forgetting about Ignis Semmelweis, and I think Alina forgot, like, hey, wait a minute. Like, did you mention him? And I was like, oh yeah, he did mention. There just isn't a slide for him, but he's in the story.
I always remember him for the pregnant woman being in the hospital being infected.
Yeah.
For his last name.
Yeah, that's a story that'll stick.
Yeah.
And then the Francesco Redi experiments.
The red meat guy.
It's in the name.
Ready.
Guy.
That's what
Exactly.
she said, Red. Did he say anything about Robert Koch?
No, I don't think we need to know that.
Okay.
No. So that was when he had the 3 flasks, and he had meat for one that was uncovered, and then he had one that had like netting on top,
Mhm.
and then
Yeah.
the other one was a completely like sealed.
Yeah, with like a cork or something. I don't know.
Yes.
Something like that.
And he watched as like in the open container there was like the worms and the stuff that were coming out of the meat, and then on the flask with the netting there were flies on top. But there was nothing growing on the meat, and then in the closed flask there was also nothing growing on the meat. So he was able to prove that I think the theory of was it the theory of bio, he was able to disprove that that life just didn't spontaneously come from non-life.
And then Louis Pasteur continued to prove that further
Right.
on with the flask experiment.
Right. And then the germ theory of disease. I think this is where Pasteur comes in.
I have to go back on this one.
So the germ theory of disease, or I think that like microbes are all around.
And causes disease. Micro causes disease.
Yeah. And there's the flask. Okay, so what this is, the 3-domain taxonomy is like you would have domain eukarya, domain archaea, and domain
Area?
yeah, I think the first one is domain eubacteria.
Area, okay.
And they have peptidoglycan.
Oh,
Yeah.
yeah. Good one.
This is true bacteria. All true bacteria is eubacteria. And then I think I have an animation for this just to do a refresher, but the second domain is the domain archaea.
They have no peptidoglycan.
Right. And this is the stuff that lives in harsh
Extreme.
environments.
Yeah.
Yeah. Prokaryotes that live in extreme environments. And then, do you guys remember the third one?
Eukarya.
Domain eukarya. Have a nucleus and organelles. So that would be like humans.
Us, yes.
Animals. And I'm going to move this just so we could see it, but gosh, I'm not going to say this right. Peptidoglycan is a special chemical on the surface, a protein sugar that defines a bacteria. So in this lecture, he says you'll learn more about that in lecture 3. So he didn't go into a ton of detail here.
Yeah, that's it's part of the cell wall, or like, yeah.
Yeah, like this.
You remember it, gram negative and gram positive, they have different thicknesses. Yeah, that's the peptidoglycan.
Which is like their cell membrane, or
Giselle, I still have more studying to do. I don't know.
The, what's it called? Bacteria also have a cell membrane. So it's just part of the cell envelope or the cell wall. It gives its shape and size or something like that. I just went over this lecture. That's why it's kind of there.
Yeah. I'm not there yet. I haven't even gone over lecture 3. I've like studied a little bit of like 1 and 2, but I'm like, ooh, I've got some catching up to do. Anatomy is just eating up all of my time.
No, anatomy is a lot.
Yeah.
But I think she got it.
Yeah. Okay. That's all that I have for this. I was hoping to get the next couple of them done, but I could not get to it. So I'm sure I will make up more slides, but for now, I'll go ahead and share my screen for lecture 2. Let me just get this up.
Lecture 2 is method of studying. Okay. Right?
Yes. Okay, pulling it up. I'll share my screen again. Okay. I'm going to put it into presentation mode so we can get it a little bit bigger. There we go. Okay. Can you guys see lecture 2 okay?
Yes.
Okay, introduction. So we're learning all about the studying microbes and why they're difficult. See the different techniques. I'm so glad we're going over this because I feel like the staining methods, I'm still
confusing.
Yeah, I think in the original lecture, I was still getting used to being in class from 8:40 to 5:30. And so I was just finding like I couldn't, like my brain was too full by the time he was getting down to some of this stuff. So I was like, I'm out.
Yeah.
So I'm glad we're going through it. So complex associations. Do you guys remember about that?
Not much.
Most habitats harbor microbes in complex associations. This necessitates separating. So
it means that in one environment there are multiple organisms living in it. As you can't ever just find one thing, right?
Yeah. sorry, I'm gonna have to keep moving this around here. The little box. Artificial conditions. Small organisms require growth outside their habitat under artificial conditions. So that's like the whole process of us having to learn what kind of environment they can thrive in, what kind of nutrients they need, that kind of thing. Microorganisms are not visible to the naked eye. Small size makes laboratory management and study of them challenging. Let's see. Okay, I think we're good on those. So what's the difference between an inoculation and inoculum?
Inoculum is the item or the colony or the specimen, like it said, that you're putting into the agar or the petri. And then inoculation is the technique, right, of like putting the inoculum into the agar to grow, I guess, to grow the material.
So the inoculum is the specimen being introduced, and the inoculation is like the whole process of the introducing the small sample.
Yeah, the method. And I think the main goal is trying to grow the organism.
Yeah, because it's supposed to like support the growth. So that's like when you're putting it in the media, you're putting it in like the nutrient agar. You're supporting its growth. So introducing a small sample to a container to support the growth. So those can be like the complex, what is it called? The complex associations, right?
Mm-hmm.
That could be, okay, I'm moving on. Incubation, that's like the environment that it needs to grow. So that would like be temperature related and like that kind of thing. Isolation. Are we good on that?
Yeah, then we're trying to separate the two different species.
Mm-hmm. So parent cells, you have parent cells here. Yeah, let's see what this says here. I was just trying to figure out what the difference between these two cells are. And I guess they're both parent cells, but they're just parent cells of different colonies.
They're like different species, I think.
Yeah.
Yeah. Each of them have their parent cell. I think that's what it said.
Yeah. And then they're just separating them here into their own individual colonies.
Yeah.
Oh, there we go. So we have the mixed culture, pure culture, and contaminant. What is the difference between a mixed culture and a pure culture? Actually, Giselle and we had already gone through this, didn't we, Giselle?
Oh yeah.
Mm-hmm.
Yeah, we just did this. So the mixed culture is 2 or more species. The pure culture is just 1. The contaminant is something that you didn't want in
There's
your plate.
There's actually a 4th bullet point that's missing.
Culture.
Okay, there it is.
Wow.
Oh.
Do you have it in front of you or did you just remember that?
I just remember.
That was one of the questions that was in the his review question. Like the
And then also when we went over this, you also skipped the last the last bullet point.
Oh, really?
Yeah.
So she remember.
Exactly.
So an example of a subculture would be like that process where you're separating out, where we did that T-streaking method, and you're trying to isolate the one culture. You're picking that one culture up, you're putting it in new media and growing it. That's an example of a subculture, right?
Mhm.
Information gathering, testing of cultures using a variety of staining techniques, biochemical tests, and genetic analysis. Identification, the final determination of the types of microbes present in the original sample. Okay. Nutrition.
The nutrients needed to grow um, the colony or the microorganism.
And then do you know the difference between macronutrients and micronutrients?
I think macro is what the majority of what we need. And micro is just a small amount that it's needed for enzyme or the structure.
And then examples of macronutrients would be proteins, lipids, carbohydrates. What else? I think it was nucleic acids is on this slide or in this lecture. And then micronutrients would be like minerals and vitamins and stuff. Yeah.
I'm gonna pull out my notes on this too, because I know that there's things here like this we need to know. 98% of a cell is composed of just these 6 elements. We need to remember carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur.
I think of chomps.
I was trying to put that too.
Chomps.
Oh.
Yeah.
Chomps.
Yeah, but it's kind of hard with the N, but chomps.
Yeah, I know when I was studying for my anatomy exam, I had like the weirdest mnemonics and acronyms. I was like, this is a very weird acronym or mnemonic that I made.
Whatever helps.
Whatever works.
Yeah.
Yeah.
Okay, so if I'm going to stop sharing, if you don't know, okay, Hoi, if you were to remember chomps, what would
Carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur.
Okay, I'm gonna try it.
Ooh.
I'm
Remember chop with an N in there, or chops with an N, or chomp.
Okay, so there's carbon, hydrogen, oxygen, nitrogen, phosphorus,
With an S. Everything's more.
and then S is
You know this one.
Smells like eggs.
Some people are
sulfur.
like this. Yes.
I was like, for some reason, silicon was coming in.
Silicon, right? I was like, I know you're
No.
thinking of that.
Silicon is SI.
Yeah.
I was like, that's not even no.
It's okay. You're good. That was good.
That's
Should we
good.
test you? I feel like you already know this, Giselle, but should we test you?
Yes.
Okay.
No, it's okay.
No? Do you remember it? You already probably remember it.
Yeah, we're good. We're good. Let's move on.
All right. The difference between a phototroph and a chemotroph.
They obtain their energy from light for phototrope, and then chemotrope obtain their energy from chemical bonding or chemical bonds. That doesn't include carbon, so they get it from inorganic molecule.
Good.
Yeah.
And the difference between an autotroph and a heterotroph. So this is just about the way that they obtain their carbon. Is that right? Am I understanding that correctly?
Yeah.
So they can obtain their carbon. So autotroph is an organism that uses carbon dioxide as its carbon source.
Yeah. So they use carbon dioxide to get carbon, and then
So they produce it within.
Yeah.
And then the heterotroph is
Us, which is where we depend on other living things to obtain our carbon, which is like we get carbon from plants releasing oxygen, because plants are photosynthetic, so they take carbon dioxide.
what does the word auto mean in autotroph?
Auto means self.
Oh yeah.
Self? That's helpful.
Like autonomic or
Me, other people.
or
Yeah.
automatic.
Yeah.
Yeah. Autograph, which is your own like graft, like your own skin, your own self.
Mhm.
Yeah,
Yeah.
it's self.
I use that to remember that they depend on themselves. Auto.
Yeah, and then hetero is different. So then they require something else, like something different for their benefit.
Yeah.
Okay, I'm just opening up to my lecture notes here because I know I've got notes on this. So, phototropes get their energy from the sun.
Sunlight.
Right?
Mm-hmm.
And then the carbon source, oh no.
Organic.
Did you say something?
Oh, never mind. They get it from carbon dioxide for photo, like plants, carbon dioxide.
So we've got, sorry, you said from plants?
The plants, they get their carbon source from carbon dioxide.
Yes.
And then do you remember examples?
Algae.
Mhm.
Trees.
Plants.
That's
Right.
right.
And cyanobacteria.
Okay. And then we have chemotrophs. They get their energy source, we said, from simple organic compounds. That's what I have here.
Yes. Then auto, meaning they get their carbon dioxide from their environment or their surroundings.
Uh-huh.
And this one is like deep sea vent bacteria. Remember, this is just one word. They're not three different things.
Which one are you talking about? Oh, you're
The
talking about
chemoautotrophs and
Oh, right.
methanogens.
Right.
Right?
Oh.
Is that what we were talking about? Or was the next one where it was purple
I
and
think-
green photosynthetic bacteria?
Yeah, you're right.
Because when we went over it, we're like, what is purple and green?
I wonder if I can, I was just thinking that maybe I will go through and like write these in for the benefit of other people so they can follow along. Sometimes it's easier, but
Yeah.
let's see. Oh, I'm gonna have to actually like write this.
Can you
Okay.
type it? Is there?
You can type it, annotate.
Where's that? Here? No. Here. See this? They're like pen color, eraser, highlighter, laser pointer pen.
Automatic. No. No.
Oh no. Guess not.
I could do it. Here. Let me see.
Do I need to make you a co-host?
No, you're good. It's okay.
Okay.
Let me know if you could see it. So then we said that photoautotrophs get their energy from this from light. So I'm just gonna put light or sunlight. Sure. Why not? Sunlight.
Are you writing? Okay, I can see
Yeah. Okay, cool. And then carbon source would be CO2.
it. Mhm.
Examples of this would be plant,
Algae.
algae, and cyanobacteria. And then, I'm just gonna mess
I
everyone
wonder what
up.
you're using. What are you using? So I know.
I'm using Zoom. So where it
Oh, you are?
says, yeah, I went to the options and then it says annotate, and then I'm annotating.
Oh, maybe I can't see it because I'm in like the slideshow presentation.
Oh, maybe.
Because I can't. Oh, wait, no.
Oh, okay.
I can do it. You see it. I'll start on the photo heterotroph. Do you want to do the chemo heterotroph, Hoy?
Here. So I started doing it. Sorry.
Oh.
And I switched the colors.
You're doing the chemo autotroph.
Oh, yeah.
Wait.
I asked Toy if she wanted to do the chemo heterotroph.
Oh, hetero. Yes. Sorry.
And
Deep sea
How do I write this? Okay.
and then we've got
bacteria.
organic molecules. Sorry, it's confusing when we're all working on something different.
How do you move it?
Oh shoot.
Can I drag it over? I can't move. I don't know how to move mine over.
Okay,
Sorry.
it's good. It's in the box enough, I'd say. And then photosynthetic. And bacteria. Photosynthetic bacteria. Oh, I remember we did that. Remember when we were sitting with Giselle and I was like, oh, this is all the same word?
Yeah, so it's actually purple and green photosynthetic bacteria.
Yeah. Just like started to put a comma there and I was like, wait a minute. No, I remember I thought this last time. That's not what it was. Okay. It should be more in the box.
How do I move it over?
That's a good question. Mine is like running into yours, but hopefully people understand it enough. So we have photoheterotrophs, get their energy source from the sun. Their carbon source is organic. Okay. I'm just trying to find some similarities here. Both photoheterotrophs and chemoheterotrophs get their carbon source from organic molecules.
Okay, that's good to know.
Photo always means sun, right? And then chemo, organic compounds.
Photo means light, actually. Sorry, I'm a nerd.
No, you're good. That's
So
why.
like photograph, it's a graph of light.
Mm,
Mhm.
okay.
And then technically, plants can grow under artificial light as well. And I just think of like marijuana. Sorry, but like they hide it, right? And then they put their lights or their lamps or whatever, so they
Yeah.
grow it under artificial light.
Yeah,
Mhm.
that's a good example.
Yeah.
There you go, baby. Apply it to life.
Okay, I'm gonna move on to the next slide. Are we good here?
Yes.
Mhm.
I'm curious to know now how I do that. Okay, hold on. I think I've got to just get out of that. Yay.
Oh no, everything's mixed up.
Can we, oh, let's see. If we go to annotate, we can clear it. Clear all drawings. Yay, look at me. I'm so smart.
Are you the host?
You're so experienced with
I know.
Zoom.
I didn't know how to annotate like 2 minutes ago, so I'm not gonna get too ahead of myself.
I didn't even know there was such thing.
So funny. Okay, other macronutrients. We have hydrogen, nitrogen, oxygen, phosphorus, sulfur. I'm just gonna see if there's any notes, if we need to know anything specific about that.
And then the last time when we were doing this with Euselin,
Yeah.
we noticed that carbon was missing from that slide.
It said other, so maybe.
Yeah,
That's
well,
right.
yeah, carbon is a very important element, I suppose. So it's not on there, which makes sense.
Oh yeah, I have it written down right here. Carbon.
Would you like to add something about that?
Like add it here.
Or yeah, I guess.
We just said it.
Well,
Yeah.
like carbohydrates, the main, like the chain is of carbon. So, and we need carbohydrates as a macronutrient. Well, as people, I don't know. I'm just making everything about humans. Sorry, but I guess for life too.
Yeah.
Mhm.
Okay, I'm moving on. Micronutrients. Yeah. I just have iron involved in respiratory proteins.
Mhm.
Other required in tiny amounts would be zinc, copper, iodine, manganese.
It's
All right there, yeah. Growth factors. What are growth factors? Those
They
are,
cannot be synthesized and they need to be provided.
yes, so those are things that you need to add to help the organism grow.
Mhm.
Then organisms are fastidious, which means that they're picky
Mhm.
and require growth factors along with complex nutrients. So this is when we get into the liquid and the solid media. The most commonly used type of media is the nutrient broth and nutrient agar.
Do you think he wants to know what's in it? Yeast, soy, beef, extract. The agar just have agar, but they both include all the yeast, soy.
I don't think so.
Wait, for what?
He wants us to know all of the components. Like, do we need to know nutrient broth has yeast, soy, beef?
The only
I don't know.
I don't.
But the way I remembered it was for broth is sappy. So SA, like I instead of beef extract, I say animal extract because I think there was one that had like sheep, I don't know,
Oh, that's
something. So I just say sappy. So S for soy, A for animal extract, P for peptone, and Y for yeast. And then for agar, because it's a solid one and it's not so sappy, it's asappy. And so the A is just agar, and then the same thing, sappy.
okay.
Sorry, it's
I
very
don't remember now
but-
because of peptone is a protein mixed with vitamin.
Mhm.
What it said.
And then the agar.
I just memorized everything. I don't even.
You're good.
An
Like, I'm
incredible
memorizing.
memory.
Yeah.
it's the flashcards. They help.
Do you handwrite your flashcards or do you use Anki?
Yeah, I use Inky.
Okay.
Yeah, so like agar, complex polysaccharide made or isolated from red algae. I know it's right in front of me. I'm trying not to look.
What's the name of the
Um,
agar? Remember he said that it's very important we know because of lab.
Oh, what is the name? I don't know.
I remember, is it start with a P?
I think it was just in like the last lecture.
It was.
Yeah, I saw that.
Actually, I
Get
think
that out
it was
now.
a part of lecture 5. I think it was it towards the end of lecture 5
Oh
or was
yes.
I feel like that might be on the exam just because it's part of
it will for sure.
algae. Where are
Is it
you?
the rhodophyta?
Oh, yeah. Start with
Yes,
Okay.
that sounds
I
right.
just looked it up.
Me too.
Used for cosmetics, food, and medical products, agar is isolated from rhodophyta, red algae.
Switching lecture. Sorry. Rhodophyta.
Yeah.
Red. Red.
Oh,
A.
here it is.
R for red. Okay.
Um, let's see. Oh, here it is. It's under the importance of algae slide in lecture number 5.
Slide 21, right? Or 22? 22.
I don't have the numbers here. I have the printouts, but yeah,
Yes.
rhodophyta. And then it says dinoflagellates cause red tides and release toxins, causing neurotoxic shellfish poisoning. We talked about that.
Okay. Good to know.
Yeah, that was a good connection. And then we have synthetic and complex media. The synthetic is the exact chemical formula.
Oh wait, to go back to
Yeah.
agar, I thought it was important, or maybe he emphasized that we had to know like the solid at room temperature,
Yes.
right? Liquefies at boiling,
Oh,
and it can't re-solidify unless it's cooled down to 42 degrees Celsius.
okay. Just going to make a note here. All right, so synthetic media is an exact chemical formula, and then the complex is components in the media that you can't, like, there's no exact science to, right?
Mhm.
Like those examples, like the blood, serum, milk, peptone. Okay. I'm also going to remember this. And then the difference between general purpose media and enriched media. Enriched media would be more like for the fastidious microbes, and the general purpose media would be like if you wanted anything to grow,
Mm, okay.
right?
Mhm. Enrich will be like a specific microbe that you're trying to grow.
Yeah, like those would be the things that you're adding to it. Like they need specific things to grow.
Mhm.
Like how we need, like how I think about it is how we need like micronutrients and that kind of thing. Like with the enriched media, you would need to add things to it. So contains complex organic substances such as blood, serum, hemoglobin, like they need special growth factors in order to survive. That would be
Mhm.
enriched media. Like I
Mhm.
just think of like enriched cereal, like you have to add like vitamins and minerals and things to cereal in order to have it be nutritious for you. And
Mhm.
it's also things that you need in order to survive. So like the enriched media versus the general purpose media. So selective media and differential media. Boy, do you remember the difference between those two?
Um, I think differential, they all have their own, they look differently. So I try to remember that because of differential. And then selective is a certain one that you want.
Right. So this one, it says, contains one or more agents that inhibit growth of some microbes and encourage growth of the desired microbes. So like selective media would be like you want one kind of microbe to grow over another kind. So you're going to be more concerned, I think, with like the nutrients of one microbe over another.
Mhm.
Whereas differential media, you are just letting everything kind of grow together. Allows growth of several types of microbes and displays visible differences among desired and undesired microbes. So this one, you like selective, you have a desired microbe, and here
Mhm.
you really don't. You're just kind of observing how things grow together.
Mhm.
Okay. As an example, I don't know if we need to know this or not.
Yeah, this one was the confusing part.
I think we
Probably should have
have to,
asked. Dang it.
yeah. I mean, I could fast forward it and play it really quick on the thing if we want to know. Do we want to do that?
Yeah, just to make sure.
Yeah, let's go to the Zoom recording really quick. I'm going to want to keep your, I hope I did the right thing. Okay, let me go ahead and pull it up because I have it up right now. Okay, this would this have been a Wednesday lecture? Let me just look really quick.
I think so.
You guys wouldn't happen to have the date on your notes, would you?
Let me see. February 7th. I think so.
The 7th?
I think so. Look into it.
Let's see. Here we go. I think this is it. Yes. I'm just trying to get it to the right spot. Growth factors. Okay, that was almost it. And so it's going to be, it starts on February 7th. And then we're going to go, it's the week after. So this would be the 9th. Oh no, it's not it. Oh, I wonder. Sorry, guys.
It's okay.
Oh, it's this one, maybe. I think it was like, this is technically like the lab portion. Oh, let me see, maybe not. Oh no, he starts lecturing about lab safety rules here. Oh. Okay, so this one is, guys, I don't know. This one.
Let me see if I can find. Does it have a time for you?
Time. Oh, here it is. Here it is. I found it. Okay, I'll share my screen. Okay, do you guys see it? Do you guys see my screen?
Yeah.
Okay, I'll move it to full screen here. Let's see if I need to back it up a little bit.
Like, so it discourages the growth of other things. Differential lets you see a lot of things, but they all appear different, so you know which is which.
I have a quick question.
Sure.
It's maybe on the scope of this class, but I guess in order to make that
Let's see if I can.
Okay, you don't have to memorize the ingredients, but
There we
you
go.
should know what the how the ingredients help you classify the media. Okay, so for example, blood agar plate has beef extract. It has peptone, which is a combination of proteins and vitamins. Obviously, it has agar, and it has sheep's blood, 5% sheep's blood, hence the name blood agar plate. Okay, based on these ingredients, would you classify blood agar plate as a synthetic or complex media?
Before he goes through, what do you guys think based on what we just talked about?
Complex.
Mm-hmm. Why?
Because we know it's there, but we don't know how much.
Right. Like the sheep blood would be like the indicator, right?
Yes.
complex.
Complex, very good. Why? Why complex?
because they composed of different. You don't know the amount of substances in it. For example, beef extract, like you don't know how much, for example.
Exactly. Yeah, exactly. So we know the beef extract is very nutritious, right? It has lots of great things, but we just don't know the exact numbers. Exactly. It would also be considered enriched as well, Megan. Yes. What makes you also say that it's enriched?
It contains things that you mentioned when you talked about enriched, like blood.
Blood. There we go. So very good. So this is a complex because it has extract and peptone. Enriched media because it has something like a growth factor like sheep's blood. Okay, very good. Yes, Hoy, it contains these very complex ingredients. Okay. Now, if you look at the plate, what do you notice about stuff growing on this plate?
There's many different types of microbes.
Not you see colonies, right? But you'll notice that in some colonies, you can actually see through the plate, like here.
Yeah, it's changing the color of the agar.
Exactly. The agar looks to have different appearances. In some colonies, the agar is bright. In some places, the agar has been eaten. It's gone, right? Because you have different appearances. Would you call this a selective or differential media?
Differential.
Differential. Anything that has a difference in appearance is differential. Blood agar plate is a differential media. It differentiates between microbes that can digest and break down the blood versus those that cannot. So these bacteria here, they're able to chew away at the blood. They can actually break open the red blood cells. These bacteria over here, they can't do that. They can't do that. Okay. This person has strep throat. They did a swab of their throat, and there is a bacteria that can break down red blood cells that causes strep throat. And so it's right here. So this person has the strep throat causing bacteria, but they also have some of the other normal bacteria found in your mouth. We have lots of bacteria found in our mouth that are able to grow on here. But here, you can tell, okay, this person does have that strep throat causing bacteria among the other bacteria in their mouth. So it's differential. It differentiates based on the ability of the microbe to digest red blood cells or not.
I have a question on that.
Mm-hmm.
So usually when people go in for a strep test, it's relative.
To get the result. Culturing requires 12 to 24 hours of growth. Okay. All right.
Do we want to keep watching this slide? Do we want to see him classify this one as well?
Sure, why not?
Yeah, because it has the starch.
We have Thayer Martin agar. Thayer Martin agar, also used in the hospitals. This one has beef extract, peptone. It has starch. It has cooked sheep's blood. This time the blood has been cooked. As a result, the blood is no longer whole. It's all melted. And that gives the plate kind of this chocolatey brown color. Okay. So the blood has been boiled and broken open. The blood cells have been broken open for the bacteria. Okay. And as a result, it kind of creates this kind of a chocolatey brown color. Okay. All right. And it has agar too. So just based on the ingredients, would you classify this as synthetic or complex?
I was trying to pause it so we can see what it was.
Complex.
Complex.
Yeah.
Why?
Because it has all these different, we don't know how much of each of these different ingredients.
General purpose media or enriched?
I said it.
I would say general purpose.
Enriched, I'd say, because of the blood still. And
right.
Yeah.
I feel like I need some time to study it because that's not sinking in for me right now.
I know, I keep having to review them.
Enriched. Right, because enriched, that's the stuff that you add in
Yeah.
The
And
definition is complex organic substance such as blood, serum, hemoglobin. So
Right.
picky stuff. So it needs to be enriched in it.
Enriched.
Enrich, yeah, so same thing. Okay. Now here's the big thing. Thayer Martin, right? Thayer Martin, it contains many antibiotics. It contains many antibiotics that inhibits the growth of lots of microorganisms, but it does allow a certain type of bacteria to grow. And that type of bacteria is called Neisseria, right? So Thayer Martin contains antibiotics that inhibits the growth of gram-positive and gram-negative bacteria. You'll learn about what it means to be gram-positive and gram-negative in lecture number 3. But for now, just know that there are bacteria that are called gram-positive bacteria. There are bacteria called gram-negative bacteria. These cannot grow on this plate. This plate also contains an antifungal that inhibits the growth of fungus. The only thing that can grow on this plate is pathogenic strains of Neisseria. As a result, it would be selective. Only Neisseria that are pathogenic can grow on Thayer Martin. So that's a very good example of a pathogenic bacteria. I mean, of a selective plate. Okay, you see how it's selective?
No,
Yeah,
no.
but I have a quick question, Professor.
Sure.
How do you differentiate between general purpose media and enriched media again?
Well, enriched media tends to have a lot of growth factors that allow fastidious organisms to grow. Okay. For example, this strep bacteria in the blood agar plate, they're very fastidious. They will only grow on media that has blood. If you were to grow streptococcus bacteria in a general purpose media, they wouldn't really grow at all. So you have to give them more of an enriched media that has things like blood in it to grow.
Okay.
Neisseria on the bottom one here, they're even pickier. They don't have the means to break open red blood cells, so you have to break it for them. And that's why we have to cook the blood, and it gives the plate a very chocolatey brown appearance. So a lot of these pathogens can be very fastidious, and you have to grow them on really precise and rich medias for them to grow.
So thank you so much.
Jimmy, I told you to email me directly, message me directly. You're spamming the chat, so please don't spam the chats. All right, you can directly message me, kind of clinically based. But yeah, there are synthetic medias. They tend to just let you grow bacteria that are just researched for like environmental bacteria.
let's go to mannitol salt agar.
Well, for the growth of mannitol salt agar, as you know, it's a complex media because it has beef extract. Oops. And it has a lot of salt. Bacteria do not do well in salt. A lot of bacteria cannot grow in environments that's very salty, right? This plate is quite salty. In fact, it has 7.5% salt. Many pathogens cannot grow in such a salty environment. The only bacteria that can grow, not the only, but one of the major bacteria that can grow in salty conditions is Staphylococcus. As a result, we use mannitol salt agar to select for Staphylococcus bacteria, right? So it's a selective plate because of its salty nature. Because it's so salty, only bacteria like Staphylococcus can grow on it. Things like E. coli, Salmonella, they would not be able to grow on this because it's too salty for them. But Staphylococcus can. So if you suspect a patient has a staph infection, you can swab the patient's lesion, grow it on mannitol salt agar, and if it grows, then you know the patient has
What's that?
staph infection. Very good. If nothing grows on the plate, then you can rule out
Staph infection.
staph infection. Very good. Very good. Well, guess what? there's different types of staphs. There's different types of staphs, right? And you can tell which type of staph a patient has because mannitol salt agar has phenol red. Phenol red is a pH indicator that makes the plate appear kind of a nice pink color, right? And the pH of at neutral pH, does anybody know what neutral pH is?
7.
7, yeah. At neutral pH, this phenol red dye is pink. It's pink. It's a nice pink color. Okay. Well, there's also a sugar in this plate called mannitol. Some staphs can ferment mannitol. Some staphs cannot ferment mannitol. When the sugar is fermented, it produces acid. Okay, when the sugar is fermented, it produces acid. What does acid do to the pH of something?
It lowers down.
It lowers it. Very good, right? If acid is introduced into this plate, it starts turning the pH indicator phenol red into a yellow color. So this Staphylococcus growing here on this side of the plate, is it fermenting the mannitol?
On the right side?
Yes, on the right side. Is it fermenting the mannitol? Yes. How about the staph on the left? Is it fermenting the mannitol?
No.
No. As a result, this plate can also be considered a differential plate because it differentiates between the two different types of staph. Here on the right, you have Staphylococcus aureus. Oops. Because Staph aureus can ferment mannitol. Here on the left, you have Staphylococcus epidermidis. This one cannot ferment mannitol. So you can differentiate the two different species of staph. Staph aureus can ferment mannitol. Staph epidermidis cannot ferment mannitol. It'll grow because they're both staphs, but only one ferments, the other one doesn't. So mannitol salt agar is both a differential and is both a selective and differential media. You can actually have both in one plate.
I have a question.
Sure.
Would you treat those two with a different antibiotic? Because they're antibiotic.
Okay. So it's selected because only the staphs can grow. Nothing else can grow besides staph. It's differential because it lets you know if you're dealing with Staph aureus or with Staph epidermidis. If a patient mannitol salt agar turns yellow, you know that they have staph.
Maybe let's go to the next one.
If there's no yellowing,
There were like some categories that we talked about because they've been salting. Is it interesting?
Yeah, like hypotonic, a purple stain. So it's a complex media because again, it has peptone, which is
Ready to go back a little bit?
a mixture
Yeah,
of
just a little bit.
amino acids and vitamins. Okay. So process, yeah, these are process plates where you put a lot of different ingredients. Okay, so here's how this plate works. Oh, I can't scroll. So it's a complex media because again, it has peptone, which is a mixture of amino acids and vitamins. It's selective. How is it selective? Well, it has this dye called crystal violet. This dye inhibits the growth of gram-positive bacteria. Here's that word again, gram-positive, right? We'll learn about this in lecture 3. Bacteria can be separated into gram-positive versus gram-negative. This one, because of the crystal violet, it inhibits the growth of gram-positive bacteria. Only gram-negative bacteria can grow. You'll learn that some examples of gram-positive bacteria are things like staph and strep.
Could you pause this real quick?
Negative.
Sorry. And go a little bit back, just a little bit, just when he's talking about how it has crystal violet.
Okay, what are we here?
Sorry.
29, 58.
Maybe like 28. or yeah, 2830. Cool.
Let's do there.
Maybe it's too much. I don't know. Thank
And it
you.
has another pH indicator called neutral red. So neutral red. Rule of thumb, if there's a pH indicator that lets you see changes in pH, it's always going to be differential, because a pH indicator can change colors, and every time you have a different appearance, it's differential, right? Differential. Okay. So process, yeah, these are process plates where you put a lot of different ingredients. Okay, so here's how this plate works. Oh, I can't scroll. So it's a complex media because, again, it has peptone, which is, you know, a mixture of amino acids and vitamins. It's selective. How is it selective? Well, it has this dye called crystal violet. This dye inhibits the growth of gram-positive bacteria. Here's that word again, gram-positive, right? We'll learn about this in lecture 3. Bacteria can be separated into gram-positive versus gram-negative. This one, because of the crystal violet, it inhibits the growth of gram-positive bacteria. Only gram-negative bacteria can grow. You'll learn that some examples of gram-positive bacteria are things like staph and strep. Examples of gram-negative bacteria are things like E. coli, salmonella, right? So gram-positive bacteria would not be able to grow on this plate, but gram-negatives can, like E. coli and salmonella. Guess what? They cause diarrheal diseases. Okay, so McConkie agar selects for gram-negative bacteria. Only gram-negatives can grow on this. Gram-positive bacteria cannot grow on this. So that tells you it's selective. Okay.
What minute was this at? Sorry.
That's okay. 3027.
3027 at 3027. Okay, so on
Here, right here, this would be gram-negative bacteria because it has that turbidity, right? Like this is showing us that there's growth. Am I understanding that right?
I guess for everything would be gram-negative. I'm assuming.
Would be too. Okay.
Yeah, because
Well, this
of the crystal violet, it's not going to let any gram-positive grow. So whatever we're seeing on that is gram-negative,
Okay.
I believe. So on the review for lecture 2, that was one of the multiple choice questions. It was number 5. An example of selective media is one that contains A, crystal violet, B, a high concentration of salt, C, various dyes to allow color forming reactions among a variety of pathogens, D, A and B only, E, all of the above. And
Wait, where did you get that?
that is number 5 on the lecture 2 review questions.
number 5. Okay.
No.
I haven't gone through that one yet.
Oops. But
Okay, I want to. Okay, I'm so glad that you said that because I did that review and I think that one I didn't know.
Yeah, I didn't know either.
so I initially put all of the above, but then shoot, I forgot who responded to me. Oh, it was Trang. Trang responded to my post and she said that she got D, which is A and B only. So yeah. Do you guys have it in front of you?
Yes.
I am looking for it right now. I can't find it. Oh wait, here is this it? Let me see.
So from this agar, the McConkie one, it has crystal violet, and we can see from the mannitol salt agar that it has a high concentration of salt. But see, various dyes to allow color forming reactions among a variety of pathogens.
I feel like it's E, right? Because it has the pH that changes color. No, wait.
Right, so that's what I was thinking, but it says various dyes, and for for like color forming reactions. You would put a pH indicator. That's
Yeah.
why I don't think it would be E, all of the above. It would be D.
It would be D because he said crystal violet is used to inhibit gram positive.
Right. And so that's A, and then B. But yeah, but I guess to allow for color forming reactions would be a pH indicator, not another dye.
Yeah, you're right. So it would be D, A, and B. Because dye is, well, yeah, dye is not a pH.
Mm-mm. That would be like crystal violet.
Yeah, might have to ask that, but it's
Mm.
good that we're talking about it.
Yeah.
Because I think I looked at it and I was like, I don't know the answer for this one.
Mhm.
Yeah. And then why would it be a high concentration of salt again?
Sodium chloride is in there.
Right.
Yeah.
So for mannitol salt agar, well, he was saying how not a lot of bacteria can survive a very salty environment, except for staphylococcus. So that's why it would be a selective media. Yeah.
Okay.
Mhm.
This is kind of a hard question.
Yeah, you really have to break it down.
Okay. This is minute 30. I'm gonna have to go back over this again. I know I will. 29. Okay. And this is, looks like February 7th. And it's the lecture at, this would be 3 o'clock-ish. That's the second half.
Wait, which lecture is it? On February 7th?
Yeah, February 7th. I think this says 1522 down here.
Yeah.
Oh,
So
nice.
that would be like 3.
Yeah,
right.
322. So the second one.
Okay. Okay. Do we want to watch any more or are we
I
good?
think I'm good because that was the last agar plate that he went over.
Okay.
So we
Yeah.
can kind of quickly review what he was talking about.
That would be great because I'm
Yeah.
still like a tiny bit lost on this. To be honest.
So first, he said that we don't need to know exactly what's in each plate, but to understand what each thing does,
Right.
right? That's what he said.
Right. Hold on. Let me see. Can you guys see the slideshow? What are you guys looking at right now?
The
Lecture.
Where it says classify this media?
Yes.
Okay. So we need to know,
the 4 of them, right?
we would need to know how to classify it based on the ingredients. So what I understand from that is he will give us the list of ingredients and we will need to be able to classify, right?
Yeah, I think so, I guess. I hope not. Shoot, that's really hard.
Really?
But
No,
yeah.
because if we know these definitions here, the difference between, right, like enriched media, general purpose media, or selective and differential, we should be able to deduct it, right? That's what he was doing with us.
I hope so. Well, okay, so let's see. So on the slide before the mannitol salt, it was the blood agar plate and the Thayer-Martin agar. Thayer-Martin?
Yeah.
Okay,
So it's
so
complex because it contains at least one ingredient that is not chemically definable. So that would be the blood. There's
wait,
blood.
are you seeing that slide? Because we see the mannitol salt and the McConkey one.
Oh, no, I'm looking at a different one. Here, I'm going to stop share. I think I probably selected the wrong one. Okay, what do you see? The types of media?
Yeah.
Okay. So this is what I was looking at.
Oh, okay, got you. Okay.
So the blood agar plate has beef extract, sheep's blood, agar peptone, which means it would be a complex media. And it's enriched
Because of the blood.
because of the blood. So it's both complex and enriched, and the blood would be the indicator for both.
Mhm.
And then the differential allows growth of several types of microbes and displays visible differences among desired and undesired microbes. And that was evident because the background, like there were certain microbes that were eating the agar and certain ones that were not. And because
Yeah.
of the discoloration, we know that it
Differential.
allows the growth of different kinds of microbes.
Yeah. Ones that digests RBCs or red blood cells and ones that do not.
See, I would think that that would be selective then. Because wouldn't that be inhibiting growth of one? If they can't digest blood, that would inhibit growth. What am I missing?
Well, in my notes, it says that it's differential because you can differentiate between microbes that can digest red blood cells and those that can't. And maybe we can't see the slide very well, but
So the growth isn't inhibited.
growth is not inhibited, no.
Okay, so that would be where I'm missing it. It just isn't digesting the blood.
Right. So maybe the way we can differentiate it is that just the discoloration, the different colors that we see on the plate
Yes.
from this picture. Yeah.
Okay.
Yeah, it's so cool. Okay, it looks like bubbles. And then Thayer Martin.
There's that indicator again where you have beef extract and sheep's blood,
This one is cooked,
right?
and that is a good growth factor for Neisseria to grow because it doesn't have the ability to break it down itself. It needs to be broken down before.
In
Yeah.
only Syria, apparently.
Yeah,
It can
only
grow
Neisseria.
later. Yeah.
That's why it's selective.
Right.
And it's also selective because there are antibiotics against gram-positive and gram-negative bacteria, as well as an antifungal, which will
Okay.
inhibit fungi. So it's really only
For Neisseria.
encouraging the growth of Neisseria.
Yeah.
Okay, and then the mannitol salt agar. We have that here.
This one is selective to only Staphylococcus because that's the only one that can grow in salt mannitol, salt agar. But then to really break it down, they added phenol red because mannitol is a sugar. And I guess to differentiate the two Staphylococcus, if it's fermented, then it will turn yellow. But if it doesn't, then it will stay the pink color.
Mhm.
Okay. I have to go back and think of the selective and differential. That's kind of confusing.
For the staphylococcus one, that was kind of confusing. But then once he said that, you know, there are different types of staphylococcus, the way you would differentiate it is with the pH indicator and mannitol. So I guess that made sense. It made more sense in my head when he said that.
Selective, but then if you want to see them differently from that one microorganism, then you have to add a pH, which will differentiate them.
Hmm.
They're both selective and differential.
Yes.
Is it? Oh yeah.
Yeah.
It's
so-
selective because it only allows staphylococcus to grow, and then and it's only differential between the types of staphylococcus.
Yes.
There you go. So that makes sense. Okay, so again, it's selective because it only allows staphylococcus. It's differential because it'll tell us what kind of staph it is.
Mm-hmm.
There you go. And then for the pH indicator, phenol red, when it's at a neutral pH, it'll be pink. And then the presence of an acid, so that's less than 7, right? Less than 7 pH
Yes.
or pH 7. It'll be yellow. And the way that we would introduce, in air quotations, introduce an acid here is when mannitol or a sugar gets fermented.
Mm-hmm.
It'll change the pH, it'll lower the pH, and then that's how it turns yellow. Yes?
Yes.
Nice.
Complex, selective, and differential. Okay.
Cool. Okay, that was really good. I'm like, yeah, it is
So
selective
glad
and
that
it is
you guys
complex. Or
re-explained
differential.
that, because I feel like I understand that maybe 5% more.
Yay!
So I'm going to try to explain it, and then
Yeah, your turn.
because I am behind the pack here. Okay. So the mannitol salt agar is complex because it contains the beef extract, right?
I think you could just say that it has all of these ingredients and we are not sure how much it is. Yeah.
Okay, so that's what
Yeah.
makes it complex.
Right.
It
Mhm.
is selective because this is only specific to Staphylococcus. And we're
Right.
trying to differentiate what kind of Staphylococcus should grow. So that's why it's differential.
Yes. So, but what
Mhm.
makes it selective?
What makes it selective? Because it only allows Staphylococcus to grow.
Right. But what's the
ingredient?
ingredient? Like why?
Mannitol?
It's because of the salt. And
Oh.
then what about the salt?
Oh, because staph only grows in,
Oh,
it can grow in the extreme environments. So only staph is gonna grow. It's gonna inhibit the growth of
others.
other things because they can't survive in sodium chloride.
Mm-hmm.
Yeah.
So only staph is gonna grow, but now you have to differentiate between what kind of staph. And the way that you do that is you add the mannitol because only one is going to ferment.
Mm-hmm.
And so you start with phenol red,
Mm-hmm.
and in the presence of acid, it's going to turn the staph aureus yellow because it's acidic.
Well, turn the agar yellow.
Right. Turn,
Yes.
Okay.
Yeah, there you go.
turn the agar yellow. So that will be an indicator of what kind it is.
Well, just think urine. Our urine is pretty acidic. It's yellow. because
Yeah.
The pH. So yeah.
So when the mannitol is fermented, it's going to turn yellow because of the acid.
Mhm.
Okay,
And then you get to see the two different type of staph,
right.
epidermis, and one is what is that?
Oris,
Ours? Yeah.
staph oris. Hey, that was really helpful. Thank you.
Yay, another 5%.
I feel like I boosted it to 15%.
Okay, there you go. Woo.
Maybe. Yeah, that was really helpful.
So then now the McConkey one.
That one's a little confusing. I still
Yeah,
process
so
it.
it's complex media, right? Okay.
Mhm.
It's selective because crystal violet is in it. So it inhibits the growth of gram-positive bacteria, meaning that it encourages the growth of gram-negative bacteria. That's why it's selective. Only gram-negative bacteria can grow.
Right.
And then it's differential because we're trying to figure out what kind of gram-negative bacteria it is.
Oh, okay.
There we go.
Yeah, I'm getting it now.
I'm getting it. Okay.
Kind of break it down really small.
Yeah. So, and then it says from the notes, neutral red is a pH indicator that allows color change with lactose fermentation.
So the yellow one will be like it's being fermented. Is that what
Oh yes. Okay. So
it is on the agar? Is that
yeah.
the
So then the red indicates lactose is fermented.
oh, the red. Okay.
That's what the red indicates. Neutral red is a pH indicator that allows color change with lactose fermentation. Oh, okay.
It's complex, but you're only desired to grow one bacteria, which is gram negative, because you don't, yeah. And then to figure out which gram negative it is, you have to differential which type by using a pH of neutral red to see the fermentation. Okay. I think
That's pretty cool.
that.
Yeah.
I didn't understand this when he went over the lecture. It was like I was so tired. I was like almost 5 or something, and I was not processing it.
Yeah.
Okay.
So I have crystal violet inhibits growth of gram-positive bacteria, but encourages growth in gram-negative bacteria.
Yes. So anything that you put on this plate and whatever colony you see, they're going to be gram negative.
Which makes it selective.
Which makes it selective to only gram negative bacteria. And so because we have like all of these things popping up on here, we still want to differentiate what kind of gram negative bacteria is on this plate. So you put a differential, or there is neutral red, which is a pH indicator, and that's what will help us differentiate what kind of gram negative bacteria is on there.
And I guess to remember that McConkey agar with the letter C in it has crystal violet, which is the letter C for crystal.
Mm.
And it only allows gram negative. C minus, I guess.
That's how I remember the positive and basic dyes.
No, I like the one that you drew for the peptide glycan, which is gram positive is wider.
Oh, you
and-
saw that?
Yes, I was like, she's on point with all this. And then the little grapes cluster.
Oh
Wait,
yeah.
Oh yeah.
Yeah, I was like, she's good. Helpful.
Cool. Yeah, right. I try, even for me.
No, it's good to have you coming to this because I feel like I understand so much more when you're explaining it.
Yeah,
Aw, thanks.
I
I
know.
try.
Now we're dependent on you.
No.
I try. It just needs to stick, right? Somehow.
That's how it sticks if you explain it, right? Like we always learn best if we're teaching somebody else something.
True.
Like I'm trying to, even in like anatomy, I'll come up to my kids, I'm like, this is your frontal, that's your mental, this is your like,
Oh yeah.
anyways, it's just, it works when you're explaining it with to somebody else, then you really understand it, you know?
Mhm.
Who was the one that said we can talk to a
True.
teddy bear or teach to a teddy bear?
Yeah, that was Dr. Dan.
Oh, someone said that.
Talk to your dog. Okay, so magnification versus resolution. We, the difference between resolution and magnification is magnification makes things bigger and resolution is clarity of an image. So it's the ability to distinguish two points as separate. Okay. I think this might be lab stuff.
Yeah, which we
I don't
kind of,
think we need to go through this. Do we?
I feel like some of it will. Probably like the staining part, you probably will ask. Um,
I am so tired, you guys.
No.
I'm just glazing over right now. Let's see what we have here. It'd be nice to go over that. But I feel like for now, like I would rather spend more time. Here we go. Positive staining and negative staining. So positive staining is, it binds to the object that you're looking at, the microbe, and it makes it appear darker. And then the negative staining makes the background darker, but the object lighter. Is that right?
Mhm.
So I'm on my desktop and I can finally show you my drawing correctly, not on a piece of paper.
Oh, good. Yay. She was
Okay,
trying to
so
show us
let
a
me,
drawing and it wasn't coming through last time.
all right, so let me share.
Oh, do I need to make you a co-host?
No, I don't think so. Oh, you cannot start sharing. Oh, okay, so
I'll make
let's
you a
see.
co-host and then you can share.
Screen share.
Oh, there you go.
Can you see? Cool. All right. Right here. Here we go. So this is what I have on staining. This was my thing.
Oh, that's really helpful.
Can you hear me? You can, right?
Yeah.
Okay, cool. So yeah, staining used for light microscopes. Cells have negatively charged cell membranes. So these dashes are the negative charge,
Mhm.
negative charges. This is the stain, the positive stain, and it's being attached to the negative charges. That's why this cell is blue.
Mhm.
And then for the negative stain, since this is a negative charge and so is the stain, it repels. So it doesn't grab on, or
Mhm.
the cell membrane doesn't grab onto the stain. So it'll just basically stain the background and then keep the cell white or clear or whatever. It just leaves it alone.
Nice. That's really, really good.
So
You scanned that in?
no, I have my iPad and then I just use Notability.
What is that? Let me write that down.
Yeah, Notability. It's my best friend. So I have my slides here too. See, I have all my notes on here. And then And then I have Anki here.
I don't know how to use Anki at all.
No. Yeah, it's pretty
I
good.
bought it, but I don't know how to use it.
You bought it?
Yeah, because I was like, everybody's talking about this. I want to know how good it is. I still didn't do well, but
Oh man. Yeah, I just downloaded it on my computer because it's free. If you just put it on the desktop. Yeah.
Did you share your Anki deck with me? I feel like you did.
I think so. It wasn't finished though. This for sure is finished. And then I also have lecture 3 now. Do you guys want to just do some of these lecture 2 flashcards with me?
Yeah, let's do it.
sure.
All right, let's do it. What are the 2 types of staining?
Gram positive and gram negative.
Mm-hmm. Good. What is differential media?
Oh God.
Differential media is
We just went over it. We're fried.
differential media is inhibiting the growth of one thing and promoting the growth of another.
Are you
Like
sure? Or
No.
is that selective?
Yes.
Oh, that's selective.
So differential media, we want to see the differences.
Between
differences.
Okay.
Yeah,
Okay.
differential media, you want to see the differences. So allows growth of several types of microbes and displays visible differences. Sorry, this is like from the slide itself, but
Mhm.
yeah. All right, we can do that again. And then what is a phase contrast microscope?
I do not know.
So then, so then there are 3 types of microscopes or light microscopes. You have what is the
Right.
most common one?
Right field.
Field?
Right.
Bright field. And then what's the opposite of bright field?
Dark.
Fluorescent?
No,
Dark
no, no.
field.
Yes.
Oh, dark.
Dark field. And then we have the phase contrast microscope. So then the phase contrast here, we'll show. I'll just show you. So specimen is contrasted against gray background for more internal detail, magnifies subtle changes. Like I just put that there too, but I kind of understand it as you can see all of the internal detail because there's like light intensity differences. But that, yeah, so it's contrast. There's light contrast, so that's why you can see more details. We'll do that again. So describe positive staining.
So positive staining is when the object itself is getting dyed because it has a negative charge on the exterior and the dye is positively charged, and so it grabs onto the negatively charged object and dyes the object itself, not the dye.
Yes, perfect. Yes, uses basic dyes or positive dye. Dye will bind to cells, which will give them color. And then describe fixed mounts. What are the pros and cons?
Fixed mount. Like the organism is on the slide. Is that what it is?
Yes, yes.
You have
So
to
how do you fix
dry
it on there?
with heat. But
Yes. That's it.
oh, those are okay. Pros and cons.
So what's the pro?
Stay on forever.
Yes.
And you can see like the internal structures of them because it's usually something that you have to, like it's heat fixed. You can like really see the like the structures, right?
Right.
Can you?
Was
I
dead.
don't know. Yeah, it's dead for sure.
That's the con. Right? When
I don't know if you
see-
can see the internal structures.
I was thinking that the one that you slice and freeze, and I think I stopped going like, no, those are the ones that you
Right.
slice and
So
freeze.
that is the transmission electron microscope.
Right.
TEM.
Yeah.
Yes. So that's an electron microscope. So then another pro for a fixed mount is that it retains its shape.
But then the con is you can't see the motility because it's dead.
Right. Yes. Specimens are dead. Cell train. But yeah, made by drying and heating the film or a film of specimen. Smear is stained using dyes to permit a visual. Oh, of cell and cell parts. There you go. Yes. So I guess you can see cell parts in cells. I don't know if cell parts means internal stuff, but maybe.
Sure, you could, because in some, yeah, you
I
could,
think you
Yeah.
could, yeah.
I think so too.
especially when you're staining it.
Yeah. Okay.
Can you expand your deck on the upper right hand corner? Do you see that box? Upper right hand corner. Can you just
All the way to the
expand
right, all the way up a little.
all the way up to the right? You see where your X is on the right?
Oh yeah.
Okay, not that. Don't do that. But
Oh, this
that
one?
Yes, 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 yes.
Oh, whoa.
Oh.
Is that better? Here, hold on. How
Oh,
about
I don't know if it's better now. But it looks.
yeah, it just goes to the
There you go.
top.
No,
Is that
it's
better?
okay. Yeah. It's just
Okay.
small.
So we'll do that one again too. What are trace micronutrients or trace elements used for?
Protein structure.
Yeah, I think so.
Uh,
It has enzyme function and maintenance of protein structure. Nice. Describe negative staining.
I did positive, so Hoy could do negative.
So the staining is negative, but our cell membrane is also negative. So they repel, so it would not stain the specimen. It will just stain the background being.
Yes. Perfect. So you're using an acidic dye, and yeah, it'll dry around it. So will appear clear and colorless on a dark background. What names are used to classify an organism by energy source?
Names
Isn't that
like autotroph.
photo?
What name?
Yeah. You're almost there, Adrian.
Photo troph something? Photo.
Yeah. Phototroph. It's just energy source. So phototroph and
Autotroph?
Chemo. Chemo.
Yes.
Sorry.
yes, chemotroph. That's it. Yeah. So energy source, photo and chemotroph.
Okay. Okay, because the autotroph is just describes like how they're getting it. They're like self-sustaining.
Okay, so then I have another question. I don't know if it's going to pop up next, but what are the names used to classify an organism by carbon source?
Oh, that's the autotroph and
And
chemotroph? No, that was this one. Autotroph.
Heterotroph.
yes,
Oh, there
heterotroph.
we go. Heterotroph.
Yes, that's it. We'll do that one again. So what is a bright field microscope?
That is a microscope that uses light on an object. I don't know. It's just the most common one. It's the one that we use in this class.
Yes. So like, what do you see? Like describe what you would see.
You would see a light background with a dark object.
Yes. Specimen appears darker on light background. We don't have to know the bottom one, I think. I just put it on there. Name 4 basic dyes.
Oh, shoot.
Crystal violet.
Yes.
Saffron.
Yes.
Something blue. And
Yes, there's a blue one.
I'm missing one. I can't think of it.
The green one. Matcholite or
Actually,
something.
green. Okay.
What's the blue one called?
Men. Methylene blue. I don't know.
Oh yes, methylene blue. Nice. Perfect.
Both M. Okay. Methylene. And
Malachite. Malachite.
I need to learn how to spell all these words.
I know that I wrote these ones down on our last study session. I remember doing it.
Yeah. All right, I'm going to do that one again. That's going to come through again soon. What is the pathway of light through a microscope? Oh, this one. This one's a long one. There are 4 steps, but
Oh gosh, I don't know.
so then first there's a condenser,
Right.
Okay.
and then
Specimen.
yeah, so then from the condenser, the light hits the specimen, and then the light goes through the specimen into the objective lens, and then I'm trying to remember. And then from the objective lens, you see an image that will hit the ocular lens, and then like there's some sort of secondary image, and then the secondary image goes through the same kind of process as the primary image, and then we'll see with our eye through the ocular lens that image. Something like that.
Wow. Okay.
But yeah, light passes
Oh.
through condenser and a beam that focuses on the specimen. Then the light comes through the specimen into the objective lens. And then you'll see an enlarged image, primary image. Then that primary image gets projected through the ocular lens. Secondary image or virtual image is also formed by the same process. Virtual image is the final image we see with our eye and perceive by our brain. Yeah, that was the slide that had the numbering from the bottom to the top. So from condenser to eye. Okay, so name 4 basic dyes.
The one that we just did?
Yeah.
Oh, okay. Saffron, crystal violet, methylene blue, malachite green, mallet.
Yes. Yep.
Hold on, wait a minute. Let me do it.
Okay.
Yes.
'Cause I remember nothing. Saffron, something blue. I can't see it. Crystal violet, saffronin. What is that? Methylene blue?
Mhm.
Methylene blue. And it's hard to read.
Malachite.
Malachite green.
Uh-huh.
Malachite green. Malachite. Is it raining?
All right. So we'll do 6 minutes for now. Okay. Name 2 acidic dyes.
I don't know these. Stop asking me questions I don't know.
Tired now. And something red. I don't remember this one.
It was Negrosin and India ink. Do I know the colors? No, but
Necrosis.
maybe we should look it up, huh? Just for curiosity. Negrosin.
Gray or black. Oh, there's a Congo red too, apparently. I wrote it down.
Really?
So Negrosin and India ink are gray or black colors, and then
Yeah.
Congo red.
Do we have to know Congo red?
I don't know. I just wrote it down.
Oh, I just got it from, where is it? Was that lecture 2? Positive. Okay, right here. Right here, Negrosin and India ink. I just got it from here.
I need to do this. This is so great. I need to do this with my notes.
Yeah, it's so helpful.
Can I get that on my laptop?
Yeah, I have it on mine.
What is it called?
Notability.
What the
Oh,
heck?
are you talking about notability on your laptop? I'm not
Yeah.
sure. You can try.
I want to do this for our next thing. Like this, that's the way to do it.
Yeah, and it's so efficient because yeah, artificial intelligence, like I'll look up differential, right? Differential, and then it'll also find my writing, and it's so cool. Hold on, let me see.
I'm
Differential
downloading it right now.
right here. Look, it highlighted my own writing. It's so crazy. It's so smart.
Wow, that's really impressive.
Yeah.
Is it hard to write on?
I'm using my iPad, so it's really easy, and I have a pen, so yeah. Then I use Dropbox, and I just upload it into my Dropbox, and then open it on my laptop or on my desktop.
I am installing it right now on my laptop.
There you go.
You can use it on the laptop to write.
I guess.
Okay.
I don't know
Try
how
it
it
out.
works.
Try it out.
We can
Okay.
finish these flashcards, but I just wanted to get that going.
Differential media.
When you're trying to find two separate specimen.
Yes.
You're trying to differentiate between the two, trying to see the
Yes.
Yeah.
There you go. Differential, differentiate the two. Yes. Phase contrast microscope. Oh, the answer's right here.
Oh, I'm not even looking at
do.
when you can see the two different, oh wait, I'm using the same word for a differential.
So phase contrast, I kind of think about it like, so then you have a bright field, you have a dark field. They're just like black and white, but phase contrast is colorful.
Oh, okay.
It is.
Yes. Oh, yeah,
Yeah.
that's a pretty one.
You can
Contrast,
see more internal detail.
Mhm.
Okay.
Specimen is contrasted against a gray background for more internal detail. It's pretty cool, pretty cool. All right. Should we see that again?
Yes.
Okay, let's do that again. Okay, and then describe positive staining.
I think I have that one down already. But
Yeah. Yeah,
that's when the
because we
stuff-
saw the picture, right? We saw my
Yeah.
picture. So
Yeah.
we're good. That was easy. What is a pure culture?
it's just one colony, like one of the same kinds of,
BC.
yeah.
Yes, contains only one species. Nice. What is a dark field microscope? Oh,
That's when you see a dark background in a light object.
yes, perfect. That's good. What are the 3 staining subtypes?
Is that gram positive? Is that, am I on the right track? No.
Mm-mm. No, those are the
Staining,
staining, yeah, but subtypes.
that's not. Oh, staining subtypes.
Right
Oh,
here.
simple stain, different stain, special stain.
Oh,
Darn.
Yeah. Boop. So simple stain requires 1 dye, differential uses 2 to distinguish cell parts or cell types. Special stain. Oh, special stain. That was when I looked it up, remember?
Yes.
Yeah. And then we're looking at like tissue or something.
sure, cell parts.
Cool. We can look at that again. Describe fixed mounts. What are the pros and cons?
So fixed mounts are done by heats. They have dead specimens. They're heat fixed onto a slide, so they stay that way for a long time, maybe forever, I'm not sure. And then you can see like the internal structures. But the cons are you can't see the motility.
Mhm.
And yeah.
I think that's it. Yeah. Fixed slides can be kept for a long time. Cell retains its shape. Specimens are dead. Cool. Name 4 basic dyes. Go, Adrian. You
remember
got it.
Safranin.
Safranin.
Yay.
I remember something about malachite green.
Yes.
Oh my gosh, that's 2. And before I had 0.
There's another M.
There is safranin. Did I already say that?
Mhm.
Yes.
Wait. What did
Oh
you say?
shoot. The answer is right here. Ah.
It's so small, don't worry.
Yeah, I could hardly see it.
Oh really? Okay.
Yeah.
I'm gonna make it bigger now, but it's not there. Can you see this? Can you see it better? Like
Yeah.
my notes?
Can you
Yeah.
put up the drawing that you drew for the gram positive, gram negatives? I want to look that up again. Yeah.
This.
Yeah. Thank you. That's good.
Mhm.
Okay, you can go back now.
Did you take a screenshot?
Maybe I should. Give me 1 second.
Sure.
Okay, there you go. Thank you.
And then you got saffronin, you got malachite green.
Okay, and then there
What's the
are,
primary
oh,
dye
wasn't there
that
like
always-
a red? Some
That
kind
was saffronin.
of- darn. I really want to say that one and have it be enough. I don't know.
What's the dye that he said is a primary one that we normally
Oh.
use?
Was that the rose?
Well, we also said that McConkey has a C because it has a certain
Dye.
So it
I
starts with a
remember it
C.
starts with a C, but I don't remember anything else. My brain is done.
I know. Don't worry. about it.
Do I say it?
No, say it.
Crystal violet.
Crystal violet. That's what I said. You don't remember?
I was like, wait.
No, I didn't say it.
And then there is a blue one. Do you remember what the first word is?
No.
Methylene blue.
Blue. Methylene blue. Something green. Malachite green.
Yes.
Saffronin.
Your favorite.
And my favorite.
Crystal.
And crystal violet. Crystal violet, for some reason, like I feel like that should be the one I should remember the most.
Yeah.
But I don't know why. Like purple's even my favorite color.
There you go. Just remember your favorite color. The basic dye.
I will just remember saffronin.
Okay, I'm gonna do this less than 1 minute just for you. You got this. What types or what names are used to classify an organism by energy source?
Chemotroph,
Mhm.
phototroph.
Yes.
Yay!
Cool. Easy. Describe negative staining. It's there. We got it.
Yeah.
Yeah, we're good on that one.
3 types of or 3 staining subtypes.
Special.
Simple.
Simple.
And there's the one that's more than 2.
The one we hate talking about, differential.
Yes. Differential. Right? It should be. Wait, this is number 2. Yeah. Differential. Good. What is a simple stain?
Only one.
That's one.
One. Describe an electron or describe electron microscopy. There are 4 things.
Really?
Oh, this is a new one for us. Okay. Even for me, to be honest. Electron microscopy.
I know they're very expensive.
Oh, yes, they are very expensive. So you could magnify it up to 5,000 to 5 million times.
It used
Actually,
short wavelength, right?
yes, it has the shortest wavelength, so you can
Yes.
see things better. Like the resolving power is something is better, or something like that. So that's 2. Oh yeah, and then we said that it was shortest wavelength, so that's 3. There was a 4th one. Let's just look at it. Beam of electron. So yeah, E minus is electron shorthand. Just saying. So yeah, waves are shorter than 100,000 times shorter than invisible light, or than visible light. Between.
I'm gonna study your flashcards. I just want to make sure that you shared them with me. But I probably can only hang for about 5 more minutes, guys.
Okay.
Yeah,
Yeah.
Sorry.
this is a very long Zoom.
I feel bad because I don't know if I can actually upload this. Like I
Yeah.
had our 2-hour one that went for anatomy. I couldn't upload it.
Oop.
So it will be like
Whoever's gonna watch it, they probably don't want to stay for 3 hours watching this.
Yeah.
Right. They'll be like, I'll just study by myself.
Or we could just say, just watch from this time to this time. I don't know.
But I think it's good because I feel like we teach each other.
Yeah.
Mhm.
So.
4 basic dyes.
Oh no, saffronin, my favorite. Crystal violet.
Your other
Oh,
favorite.
you got it.
Yes.
And oh no. Blue, something blue, right?
Yes.
What kind of blue?
Methylene.
Methylene blue.
Yay.
Hey, I'm getting better.
Acidic dyes.
Oh no.
Gross.
Oh yeah, nigrosin.
Mhm.
India ink.
Yes. Perfect. What names are used to classify an organism by carbon source?
Autotroph
Mhm.
and
Yeah.
heterotroph.
Yes. What is agar?
Nutrients solid broth. No, nutrients solid. They can't be a solid and a broth.
Yes, right. So this is the reason why I remembered. So it was a complex polysaccharide that is isolated from red algae, which we said was shoot. What is it?
Something.
What is
Oh, is that red? Yeah.
rhodophyta?
Okay.
Rhodophyta. It is solid at room temperature. It liquefies at 100 degrees Celsius, and it cannot cool down it cannot re-solidify unless it's cooled down to 42 degrees Celsius. And it is normally not digestible by microbes, and it holds moisture and nutrients.
Yes.
Shoot. All right, that's that. What's a differential stain?
You're trying to differentiate the two type.
Yes. Uses 2 dyes to distinguish cell types and or
Oh, the
parts.
stains, 2 dyes. Okay.
Mhm. Good. What is a phototroph?
Being sunlight,
Yeah.
right?
Yes. Energy from light. Good. What is a chemotroph?
Energy from carbon. Sorry, go ahead.
No, that's okay. Go ahead.
Energy from chemical bonds. Inorganic.
Yes. What is a special stain?
Looking for cell structures. Right?
Yes. What is enriched media?
Nutrients that you put inside a media to make fastidious organisms grow.
Yes. Contains complex organic substances and special growth factors. That's how I remember it, because of the fastidious microbes. Anyway, what is a heterotroph?
Heterotroph is getting nutrients from outside sources.
Yes. Obtains carbon by other living organisms dependent on other life forms. What is a phase contrast microscope?
Phase contrast microscope. That's not the light one, is it? Where you can see 2 different kinds of light and you can see the internal structures better.
It is that one.
Yeah.
Yes. What is synthetic media?
It's a specific amount of organic material that in there.
Yes. So what's the opposite of synthetic media? I mean, material, synthetic media.
Enrich, right? Complex? Oh.
I think it's complex. Yeah,
Complex.
enriched and general purpose is the one. Right. selective differential, yeah, general purpose enriched, and then synthetic complex. Cool, nice. Universal recipe, complex media.
But it has many ingredients.
Yes, and we
Yeah.
don't know what's in it or how
Yeah.
much of it. Nice. A cell composed of what 6 elements? Remember?
A cell is composed of so a nucleus.
No, it's chomps. 6 elements.
Oh, okay.
The elements.
Carbon,
Mhm.
hydrogen, oxygen, N, that's weird, nitrogen,
Yes.
phosphorus,
Yes.
Huh?
and
Your favorite?
not silicon. I'm not gonna say that.
But
What is it? S.
eggs.
Oh, sulfur.
Yay! Job.
Nice. And look, I even put chomps. See? All right. What is a cell culture? We're past your bedtime, Adrian.
I'm
Yeah.
By a
sorry.
minute. Sorry.
I am like turning into a pumpkin over here.
All right.
Thank you so much for your guys' time. I really appreciate it.
No, thank you.
you.
Yes, and thank you, Giselle, for like helping us out. I was
Yeah.
course.
I actually like doing this kind of stuff better. I feel like the PowerPoints are good to kind of get like an overview and test questions, but I really appreciate this too.
Cool.
I really like this.
Yeah,
So.
it's fun.
Yeah. Oh,